So Andrea has recently joined us in Harbour at the start of October as a group leader. She did her PhD with George Sheldrick in Göttingen. So the crystallographers amongst us, that's a known name. After which she went to the Cambridge to work with Randy Reed and Garib Morshidov, really trying to push the limits of what's possible in a crystallographic experiment. Then she came and I managed to snag her for a brief postdoc in Hamburg before she went down to Würzburg to carry on working there. And in Würzburg, she launched her own group. And as I said, since October, she's been with us in Hamburg. And as a methods developer, one of the things Andrea is very good at is understanding the limits of structures. And when coronavirus hit, she pulled together a collaboration of structural biologists from across the globe to really try and make sure the structures that we as a community are delivering to our downstream users, to clinicians, to drug discoverers, are the best they can be. And I think that's what she's going to talk to us about today. So I'm going to hand over to Andrea. I'd ask you all to keep your questions to the end, but you're welcome to put questions into the chat. And then hope you all enjoy the seminar. Andrea, take it away, please.
It is quite an honor to be invited to speak here, in particular, so shortly after coming to Hamburg. As Arwen said, she snatched me away, and now I'm going to talk about what we do with the coronavirus, or what's inside the coronavirus, and what the limits of what we can see are. But first, I'm going to give a rather general introduction for any students and maybe among the audience. And that is some fundamental questions. How big is the virus? So this is a human hair electron microscopic picture, and you can see that the hair is a little bit less than 100 micrometers in diameter, perhaps 30 micrometers. And this is the virus in comparison. And if your screen is not very large, you may not be seeing the single red blob. So we're going to zoom in, and then the virus becomes somewhat visible. So this is 150 nanometers. And the problem is that if the virus is 150 nanometers and an atom is 0.1 nanometers, in order to make a picture of the virus, let alone seeing the atomic structure of the virus, visible light doesn't cut it. Because visible light has a wavelength of 600 nanometers or 400 to 780, but this yellow one is 600 nanometers. So there we go, that's the problem. And what do we usually use to solve this problem? That's right, large facilities. So we use X-rays, which are 0.1 nanometers, and also neutrons, about which I'm not talking so much today, because they haven't been used extensively in coronavirus research yet, and electrons, which are 0.003 nanometers. So with those two types of photons and electrons, so we could potentially, theoretically, make these structures visible, and that is what we do with methods like X-ray crystallography or single particle cryo-EM. Here's a transmission electron microscopic picture of SARS-CoV-2. So as I said, electrons are able to give us these details. And of course, the virus has no color, but we like to color it to make it look a little bit more warning, dangerous-like. What you also can see nicely in this picture is the corona around the virus, which looks like the blackened out sun during a solstice. And this is what where the coronavirus got its name from. There is also SEM pictures, which are, I think, strikingly beautiful. This is some lung tissue, so you can see the cilia, which are usually clearing away dirt and spores and dust and whatever you breathe in from your lung. But in this case, they are heavily infected with coronavirus, which can be seen all over the surface. And then the long slimy bit you see here is, you wouldn't have believed it, mucus. So this is slime. But we can visualize all these things thanks to electrons. But of course, we would like to see more detail. So actually, the virus is large for a virus, but it's still pretty small compared to like bacteria, for example. That allows us to see almost the entire virus in atomic detail. So how small is an atom in comparison to this virus? So we take one of the spikes on the surface. The spikes are, by the way, what makes those nice corona. cut it out, move it to the side, and we make a more schematic picture. These pictures are from Thomas Spletschlöser, our illustrator in Berlin, who's really making beautiful works. So this is one of the spikes in the surface. And then if we zoom into that with, for example, Pymol, you can see the ribbon model of the molecule inside. You may all be familiar with those. And then we could, of course, change the ribbon molecule to a stick model of the actual single atoms, but then the whole spike would become really difficult to picture and would be difficult to find things. So we are usually referring to these structures either as a ribbon model or as the surface. However, in order to get those, we typically will have an atomic structure. So yeah. But where do we get those from, right? Where do we get those atomic structures? An additional problem is that the coronavirus is stoichiometric in its composition. So while small viruses like the rhinovirus has a hard protein shell, so theoretically it can be crystallized. This is not true for the coronavirus. The coronavirus has a dilipid membrane. And the spikes are not ordered, they're floating in this membrane. The other proteins as well. It can vary between 50 nanometers and like 250 nanometers in diameter. It's more or less like a bubble that averages to be 150 nanometers. But we cannot, we cannot kind of like do atomic resolution structures of things where we cannot purify large quantities of similar particles or molecules. So we cannot have an atomic structure of the entire virus, but we can make atomic structures of individual parts of the virus. And that's actually already pretty helpful together with those EM pictures I've shown. But where do these structures come from? So first of all, there are 29 proteins. So together with the RNA, there are roughly, depending on a little bit on how you count, 30 macromolecules. The RNA inside the virus, and 29 proteins, of which 4 are called the structural proteins, which are present in the virion, which is the thing that gets depicted everywhere. That is the spike, the envelope and membrane in the hull of the virus, and the nucleocapsid, which is packing the RNA inside the virus as an additional layer of protection for the valuable blueprints of other proteins. Then there are the so-called non-structural proteins, of which there are 16. And those 16 are only produced once a host cell has been infected. And then there are 9 accessory proteins, which are also only produced once inside the cell. But we do know very little about their function, and very few structures have been solved. Out of these 29 proteins, as of today, I think we know 17, at least in part. So there is still a big room for new protein structures when it comes to corona. There are in total 846 structures. This is today's number. On Wednesday, we always get the new structures. So this is last Wednesday's number. And most of those, 638, come from X-ray crystallography, of which, by the way, 1 is a combined neutron structure. These X-ray crystallographic structures cover all 17 proteins, except for RNA polymerase, which only has been solved by cryo-EM. There are 32 structures from nuclear magnetic resonance. And these cover leader protein, which is the NSP1, the first non-structural protein, envelope protein, and 6 others, and 177 cryo-electron microscopy structures, which are to a large extent spike and RNA polymerase structures, and have told us a lot about how the spike works and how the RDRP works. So where do they come from? Those 638 structures mostly come from synchrotrons. Some 56 come from Holmes-Force, and 4 come from X-Fells. Those 576 still make up the majority of structures that are known from coronavirus, basically showcasing the importance of large facilities when it comes to coronavirus research. We would not be where we are today with our knowledge of life science and our design of drugs if it weren't for X-ray crystallographic structures from synchrotrons. And as you may know, within Hallos, there are a number of these facilities. The synchrotron next door to us, Petra 3, at the DESY campus, then an XFEL source, the European XFEL, and I'm hoping soon to see a few coronavirus structures from the XFEL. ESS is a neutron source, which is still being built, which is possibly why we don't have so many neutron structures yet. And MaxIV, which has also contributed to the pool of coronavirus structures. So, given we are living in this beautiful new world of over 800 known structures, one would think that is a golden age. Like, we're having a pandemic, but at least we have those great tools to know viruses at atomic resolution. And it is true. I mean, we would not be where we are today if it weren't for the international collaboration of researchers and the tools we have in 2020. Even like 30 years ago, this research would have been much more difficult, if not impossible. In particular, when it comes to cryo-EM, I mean, many things would just not have been possible. If you want to solve a structure with crystallography or with any other method that is NMR or cryo-EM, you always need a model. So in none of these like free methods, NMR, crystallography, and cryo-EM, are you ever getting a structural model out. You are always interpreting your experimental data with a model. So the model is your hypothesis. It's not the direct outcome of your experiment. And without a model, you can't get anywhere. You cannot get your answers to biological questions. And in coronavirus, very important, you cannot do downstream methods such as structure-based drug design. However, there are some problems which look like this. So either we solve a structure, but we cannot see everything we want, or we solve a structure and it's parts of it are wrong, or most devious of all, we think we solved the structure correctly, but actually it was so wrong that the interpretation, the answers it gives us are wrong as well. Where does this come from? So if you measure X-ray data, you measure amplitudes or intensities from which you calculate amplitudes. And those amplitudes can only be translated into an electron density map as the one displayed at the bottom here, if you also have phases. And these phases come from your model. So in a kind of circular process, you are improving the model with the map and then reinterpreting your amplitudes in order to get a new map. And this, of course, leads to model bias. So you cannot observe what you don't expect. Here is a typical electron density, and it has some defects. Some of the model is not covered. So building a model in here, in this map, might be slightly difficult. What we would like is a map like this, where the model fits rather good. I mean, this is, of course, a low resolution map, but the model fits it well. That's what we would like to have. We measure this fit between model and map in crystallography with an R value, which gives us the discrepancy between the measured data and the model structure in the terms of like FOPS, which is observed, and FCalc, which is calculated from the model. And in a typical map like this one, that's 20.4%, but we would like 0%. So we would like a perfect fit between model and data. Typical R values for small molecules are in the range of 5%, while for macromolecular structures, they're more like 20%. And this constitutes actually one of the fundamental problems my work group is dealing with when we are not researching coronavirus. The question, why do our models not fit the data? Either the data quality is bad, or we're missing something in our models, or the optimization between map and model isn't perfect. So this also makes interpreting these densities really difficult in crystallography. Well, cryo-EM has its own problems about which I'm not talking today, but there are others. And now we add to all these problems of interpreting the experimental data with a model, when even a very good model doesn't fit the data better than, say, 80–85%. Now we add to this the pressure of a pandemic and the pressure of publishing in high impact journals at a high speed. The results are even more errors than usually happen. And this is reflected well in the publications that have come out. So there has been this one which claimed that HIV and spike have similarities. There was this early remdesivir study, you may remember, which has problems with its statistics. And then there is, of course, this energy calculation, which was an MD paper. And instead of taking the lowest energy conformations they observed in MD, they took the highest energy conformations, which is really possibly just an outcome of being under too much pressure and not having gotten into the topic deep enough. And I'm very far away from making any accusations of a personal nature here. The reality is that even in a normal environment, errors happen in every structure. I think there is errors in each and every structure in the PDB, or things that could be better. But now add to this sitting alone in home office with a difficult access to experts and colleagues, having to watch out for your children, having the high pressure of publish tomorrow. The structure needs to be refined so we can make the pictures and publish this. And of course, errors happen. So we need to watch this, right? We need to do something against it. But the problem is that once a structure has been pictured and published, the authors usually have very little interest in improving it. And under the current circumstances, it seems that almost every paper that say structure coronavirus can get published. This is why we started the Coronavirus Structural Task Force, to look out for these structures. We are 23 people from 7 different countries, and we are across 9 time zones. So in the beginning, of course, we were not 23 people. We started out with my lab. I asked my postdoc union if he wants to join. That was the first person I asked. And then I asked my students, and then we asked Gianluca Santoni from ESRF, and then we asked Tristan Kroll from Cambridge, and then we got contacts, and after a few months, we were 23 people. Now, most of us are methods developers in structural biology, except, the web designer and the media designer. And that means as methods developers, we are uniquely suited to this task because we are specialists in solving structures and expert users of our own software. We are also relatively confident to point out, say, chemical or usage errors in software and in modeling, or processing problems in the data processing, because, well, we must be more familiar with the methods than other people because we develop them. We are evaluating all published structures from SARS-CoV and SARS-CoV-2 and some selected human interaction partners such as ACE2. In some cases, either because it's a representative structure for that protein or because it's of high relevance to drug development, we manually look at the structures as well. So not only do we an automatic evaluation, but we also manually look at these structures. And in some cases where we find errors, we will also put online an improved structure. And we, of course, first of all, go back to the original authors who did the pioneering work of solving the structure in the first place. I mean, this is really the breakthrough. And we tell them right, your adenosine needs to be the other way around. Would you care to correct your PDB entry? Or if they are still in a preprint stage, they're often very thankful for us to point this out because they can also correct it in a publication. So during the last months, we've caught quite a few of problems before they had been published, which made us proud, but is a little bit difficult to quantify. We also do publish these data in a GitHub repository, which is accessible via our homepage inside corona.net immediately. So as soon as we're finished with like remodeling a structure, or as soon as the new PDBs come out, we run everything and we post it online immediately. That also makes it a little bit hard for us to know who our downstream users are exactly. So we can see all the downloads our GitHub has, but we don't know who they are, unless they write to us, which some do. Some 70 groups have written to us asking advice on different topics, like which structure should we take for our problem and our modeling. But of course, most of them are not pharmaceutical companies. So we know that we know that the pharmaceutical companies are using our data as well, but we don't know who they are exactly because they don't talk to us. And we do literature reviews for scientists to highlight certain proteins from a structural point of view, essentially giving a literature review and also addressing topics on how to choose the correct structure, for example, for molecular dynamics, and annotate all the structures. And we do some public outreach about which I'm going to talk in the end. This is the banner of the homepage. As you can see, we have for the public and the virus, which used to be for scientists. And you can see many different blog posts, but we also have some database information and so on. So what do we do? On Wednesday, the new PDB entries come out and we download them, and then we collect metadata in a SQL database, which is a dedicated database we programmed. We compare them to the NCBI proteome of SARS-CoV and SARS-CoV-2 in order to classify, is this structure, for example, E protein, which you may think is easy, but it's not, because host organism is not always given as the virus in the PDB. So actually aligning the entire PDB, every new entry onto the protein, is the safest way for us to find actually bits of SARS-CoV-2 and SARS-CoV. If there are raw data available, which is unfortunately mostly not the case, we also get those raw data. And then we do RMSD calculations between the new structure and all structures of that protein that have previously been solved, which tells us, for example, is this a new overall fold? We also do a sequence alignment, which tells us, like, has this bit of the structure been solved before, in order to get some kind of idea. And we will also discuss the new structures in short every Wednesday. Then we do validation with programs like Phoenix Triage, check, and PDB redo. In order, PDB redo refines the structure. Phoenix Triage analyzes the data quality. Check does a little bit of both. Phoenix more probity and Phoenix Clash Core are used in a special dedicated way because the more probity server is currently seeing so much load from people who develop coronavirus drugs that we decided to have a dedicated PDF for each structure, giving the more probity stats, so people don't have to rerun the same PDB entries through more probity. This is called more probity Rama chart. Then we use the programs Auspex and Haruspex, which are developed in my group. Auspex is a diffraction data analyzer, and Haruspex is a neural network to predict and annotate secondary structure in cryo-EM structures. We use a glycosylation analysis program, NetNGLYC, and we use, of course, the CCPM validation pipeline, which gives us an overall idea of cryo-EM data quality and cryo-EM modeling quality. Then we go to a manual step. So every structure has the Auspex plot looked at, because our machine learning project for that is in development. And so we look at each Auspex plots. And for some selected structures, we also do reprocessing with XDS and re-refinement with Isolde, RefMac, or CCPM, depending on who does that. So Tristan, of course, uses his own software Isolde, and Agnel uses CCPM. And then Semhoral and I use RefMac. And if we need to reprocess data, we are very lucky having Kai Diedrichs on the task force, who will usually do that kindly for us. And then we disseminate it on GitHub. We also tweet. We write about new proteins on Proteopedia and on the homepage. And 3D BioNotes also allows you to see all our structures. And downstream users, as I said, many individual labs that have contacted us, many we don't know about. These are some we know about. So MolSSI directly interfaces into our database in order to have at their COVID resource for modelers up-to-date information. Folding at Home has done a lot of their huge calculation effort on our structures. The JEDI challenge, so this is the JEDI Grand Challenge, 1 billion molecules against COVID-19, which was a monetary prize for in silico finding a good drug candidate against COVID-19 on selected targets. They recommended that our structures be used as the starting structures and that modelers critically assess our validation metrics to find out what the best models are for them. And lately, we also found out that IBM Open Pandemics has also been using our structures from the start, which I thought was pretty cool. But as I said, because they're online, we often don't know who our users are. So if you are, please let us know. We're interested in knowing. Here's 2 examples from the software developed in my group. One is for HARUSPECs. So this is a very nicely resolved 2.8 angstrom cryo-EM map from the spike head. So if you have the spike, this is the spike, I hope you can see me. Yeah, right, this is the spike coming out from the hull. The bit we're looking here is the top head, right? It's also interesting because that's the bit that the antibody binds to. So if you are vaccinated, you'll have that antibody and it binds to the spike. So, however, if you start modeling the structure, you want to validate it, you may want to know where are the beta sheets and the alpha helices. So if you run HARUSPECs over it or download HARUSPECs results from our repository, we give you something like this, where red is helices and blue is beta sheets, and then you can compare to the model. There's also a paper which has come out. The other thing we do is Auspex. Auspex is also available over a web server, Auspex.de. This is a structure from the main protease, and what it does is it does a plot, in this case, of FOB, so that's the amplitudes, the diffraction amplitudes, versus the resolution. And each dot is one reflection that has been recorded. What you can see here is a spike in the distribution, and that stems from an ice ring. And if you actually look closely, there are more spikes here which haven't been flagged. How do these spikes come into being? Well, they all occur at the same resolution. Of course, these are ice rings. So there are ice diffraction from many ice crystals that formed on the crystal during measurement or before measurement. And this ice contamination structure. So this spike is actually standing for some biasing of the data that we don't want. But at least now in deposited data can make visual. Because as you may remember, an ice ring, if you're a crystallographer, you know, ice rings are easy to see on a diffraction image. But the diffraction images are mostly not deposited because they're not mandatory in the PDB. So how can we discover them in MTZ files or in ZIP files in the PDB? And Auspex is the answer. Auspex helps you to find those if they're and we found ice rings in this way in 15% of all crystallographic data sets from SARS and SARS-CoV-2. So 15% could be possibly better with reprocessing and could have used some cryo optimization, so some better cryo protection during cooling the crystal. What else did we find? In crystallographic data, most data sets, except for, had a good completeness with over 80%. The signal strength was good in all data sets. That came a little bit unexpected for us. We actually expected to see some data sets that were really weak, but none were. Potentially twinned due to L-test, 11.8% of all data sets. But again, we could do nothing against this except twin refinement during refinement, unless we have the raw data, in which case we can do a better job at it. Problems with the beam stop mask. 11% of data sets. So it seems that many of those synchrotron beamlines are not doing a very good job in actually masking out the beam stop. And when I last talked to Kai, we are actually wondering, because it would be very nice to have an automatic way to find the beam stop mask. In particular, during the early stages of structure solution during phasing, having low resolution reflections that are wrong, close to zero, because your beam stop mask wasn't good, can really hinder you from solving the structure. I actually have a few examples more. If you delete like the innermost 10 reflections, the structure solves, but not very well. And if you actually integrate the structure correctly, it becomes a lot better. Only 2 structures had really bad R values, meaning a very bad fit between model and data, and that were 2 structures. But in many structures, details of the model were wrong. I'm going to address this in the next slide. As for cryo-EM data, we looked at the average model Fourier shell correlation, which is something that is somewhat comparable to an R value, and a mutual information score, MI. 7% had a too small average model FSC, and 9% a too small mutual information coefficient. So we would say that 7% had a bad fit between model and cryo-EM reconstruction map. 15% of structures in cryo-EM had more than 5% of the residues fit poorly in the map. And we calculated this with the segmented Manders overlap coefficient. So, you know, some residues always don't fit in a map very well, but more than 5% is somewhat worrying. And it would be good to not look at each and every structure manually, because it's just taking too much time. And the task force is with 23 people, quite a few people, but not enough to look at all of these structures, let alone that in order to do corrections to structures, you really need to know what you're doing, because you cannot possibly go back to offers having made a structure worse in any respect. So you need a postdoc to do this, or at least a very experienced PhD student. 85% of structures had a relatively good density fit and were overall statistically sound. In modeling, interesting, there we are. More probability results can inform modelers in general, and that's why we have this dedicated special summary PDF, about which I'm not talking now. But one problem we have is that the Ramachandran plots are often not helpful anymore, because in particular at lower resolution, people are now using Ramachandran restraints. And as soon as you have used Ramachandran torsion restraints in order to make your structure adhere to the Ramachandran plot, you can no longer use it as a validation criterion. So we need something different. And what we are using is Kablam score, which has been published by the Richardson lab at Duke University, who are also members of the task force. And using this Kablam score, which evaluates backbone torsion over 7 residues, as opposed to Ramachandran, and is not used for restraints, 31% had more than 2% outliers in Kablam scores, which means 31% had potentially incorrect backbone conformations in some larger parts of the structure. And an incorrect backbone conformation, meaning that the amino acid main chain is somehow twisted or not like where it should be, means that you really need to revise how you build the backbone, because it may be a wrong fold. We also saw, when we did manual evaluation, which was first done by Tristan Kroll, and with others joining him later, like Dale or Sam, we saw peptide bond flips a lot, uncommon rotamers, of course, which are easily addressed. What we also saw surprisingly often were messed up cysteine zinc sites. So many proteins in coronavirus interact with zincs, and we did actually are currently doing a statistical study on this. what happened very often is that people put a disulfide bond where the zinc should be sitting, or that if there was a zinc, about 90% of them had a messed up geometry, meaning there was not like a regular coordination and the bond distances were too long, too short. The angles between the coordination partners were not correct. Misidentification of small ions also happened. So that include, for example, and the multi-zinc side modeled as PEG. So these happen fairly often. And then N-linked sugars, glycosylations, were flipped the wrong way around the asparagine. And this happened in about, I think in the beginning we did a plot, it's a long time ago, so I don't know if it's still true, but in the beginning there were about 50% of glycosylation sites. And we don't know why. We have researched with people like Paul Emsley to find out whether COOT is kind of like generating the problem, and this seems not the case. We don't know why the asparagine, so why the sugars always end up belly up, like flipped the wrong way. And that, of course, then in consequence means the rest of the sugar chain has a bad geometry. I'm going to give two examples in more detail. One is NSP3, which is the largest of the NSPs with 1,900 amino acids. It has 15 different segments, and it excretes RNA into the cytosol, as our colleagues from CSSB found out recently. But here we are focusing on its other function. There are many functions, but these are the two which have been well known, and that's the cleavage of the first 5 NSPs from the polypeptide chain that is produced once the virus starts making its protein chain. It makes one long chain, and then that has to be cut into functional NSPs. And that's the papain-like protease that does that. It cuts off the polypeptide chain into functional bits. So if you could interrupt this, you could stop the coronavirus from reproducing, basically. And then that makes papain-like protease a popular drug target. It's actually, I think, number 3 on the list of like popular drug targets in coronavirus. And there is zinc fingers, and a zinc finger in the protein is essential for cleavage. So the first coronavirus 2, so new coronavirus papain-like protease to come out was 6W9C, released on the 1st of April. Has a resolution of 2.7, an R-work of 23, an R3 of 30, a bit high. Overall completeness, however, was a bummer, 57%. So how did that happen? Luckily, this was from a structural genomics group. So the raw data were deposited in proteindifraction.org, which doesn't happen very often. So we had raw images. And we looked at them and we found out they did 30-degree measurement, followed by 60 degrees starting from the same angular position, which is a very curious strategy. And I suspect that they didn't think that this crystal was like what they wanted, or that something went wrong here. We tried to communicate with the group, but they stopped answering our emails, unfortunately, because I would really like to know how that happened. And I'm sure there was an error of some sort, but it could also be that their measurement software kind of messed up. It would be nice to go to the bottom of this for the sake of improving the strategy. The angular range per image was too large, in my opinion, 0.5% on a dextrose Pilatus detector. The fraction was highly anisotropic, so the threefold NCS made the structure solvable despite the low completeness. But the zinc fingers had been modeled differently in each copy, with the zinc missing completely from one. We took that data, which, as I said, were really important to the drug developers and was the first one available for this protein. Kai Diedrichs did a reprocessing in ASDS, omitting the final images of both the 30 and the 60-degree sweep, running star aniso in order to get an elliptical resolution cutoff to address anisotropy. Resolution improved to 2.6 angstrom, with a better data quality overall, but an even lower completeness, 44.5%. However, the data that we retained were better in quality. Then Dale Tronrod went on to model this in Oregon. He added zincs to all sides. He restrained them properly to be correct in their coordination geometry, used NCS restraints between the 3 copies, and a higher weighting of the ideal geometry in the face of the low completeness, reassessed the side chains and the waters, and hence dropped R3 from 30% to 25%. So that's a 5% R value improvement and a much better view on the zinc sites, which are still really ugly. But this structure was now a better starting point for drug developers. Since then, more papain-like protease structures have come out, and they had full completeness and were better. But at the time, for a few weeks, this was the only structure that was there. And we hope we could contribute something on like making drug development based on the structure better. The other example is RNA polymerase bound to remdesivir. Here's just quickly, because I think it's beautiful, an example from Haruspex again. So what you can see here is the RNA is here, and the RNA is golden in Haruspex. And this is the Haruspex results, which nicely segments the map into beta sheet, alpha helix, and nucleotide. But let's look at the structure in some more detail. Oh, that's the model. I'm going to show that again in a video in a moment. And RNA polymerase is the copy machine of the RNA. So its job is to make more RNA for future viruses from a template strand. And it is encoded in the viral genome because the virus likes to bring its own copy machine in the cell, and then it forms from the endoplasmatic reticulum some kind of foam in which the RNA is produced for new viruses. Without it, new viruses cannot be made, and the cycle of infection is broken. So it is a viable drug target. And a well-known drug has been used in Ebola to address the RNA polymerase of the Ebola virus, which is remdesivir. Remdesivir is a nucleotide analog, and that means it looks to the body like adenosine, which makes up RNA. So this doesn't look like adenosine very much, so I wondered a little bit. Turns out it's metabolized in a rather complicated process, ending up with something like this, which looks a lot more like adenosine to me. So this is the reason why remdesivir doesn't look like adenosine. And it kind of reassured me to see this as a chemist. So RNA polymerase thinks remdesivir is a nucleotide and therefore kind of like wheels it in because it looks like RNA to it. And once remdesivir is bound to the nascent chain, opposite the template chain, it jams the whole copy machine, and the making of new RNA is stopped, and the RNA polymerase is blocked. It cannot produce more RNA anymore. And this is how it works. This is how it affects the body. And this has been that after working in Ebola, it would also work for SARS-CoV-2. In order to fight COVID-19, it could be given to patients. Now, the WHO and NIH are on like different opinions here, and I'm not going to meddle with them. But one indication that this may be true was, of course, the coronavirus RNA polymerase bound map from that cryo-EM structure. And this is the model that was built into this. And what you can see here in green is the template strand, and in orange is the new strand. And then in purple, bound at the end of it, is remdesivir. Now in this structure 2BV7, the authors originally had also included a diphosphate and 3 magnesium ions. And we were wondering how well they were supported inside the reconstruction density. So because if you show the entire protein, you cannot see very much. I'm rotating it here a little bit so you can see the remdesivir clearly, and also the magnesium ions and the diphosphate. So one reason I think they put in magnesium is because it's been postulated for the mechanism. So here is the density, and as you can perhaps see, it is not like very well covering the free magnesium and diphosphate, which is why we think there is actually no magnesium and diphosphate, which may make a huge difference to people who want to design a drug based on this. So we actually managed to catch this at an early stage, and the structure in the PEB has now been revised. They left in one of the magnesiums, we don't know, possibly they had other biological evidence for, but we couldn't see any evidence for the magnesiums. Another thing that happened with this structure and with every other RNA polymerase structure up to that point was there was a 7 and 9 residues out of register shift in the C terminus, which can be corrected in Isolde pretty straightforwardly. And this is a video showing it from Tristan Kroll. So every SARS structure of RNA polymerase and every coronavirus structure up until that point had that error until we found it, because they'd all copied each other. And then we could correct it, or Tristan could correct it. And here you go. Isn't that nice? So Isolde is doing molecular dynamics while you are like fitting your structure. It's a new approach to modeling. And I think it's really nice to see the whole structure wobble while you are correcting it. And then he corrects a few little bits and looks a little bit at the density, and now the chain sits in there very nicely and the side chains are all resolved. So the problem is the first SARS structure from RNA polymerase really had no side chains visible. You were not sure where the register was, but later showed a density, and that clearly was not fitting the side chain pattern. So we also found an adenosine base that was more the phosphate like this, it's set to the phosphate like this, and we don't know why. And 2 cis peptides and a few, we could find a few more waters. So this was a fairly typical like thing we did in the end. And we, of course, immediately put the structure online. With that, I'm almost at the end. I just want to show you a few cool things we also did. We did a life cycle. There will be an animated version available, I hope, next week, together with the University of Utah, where you can see entry, release of nucleocapsids, formation of polypeptide chain, then the foam-like thing for the production of new RNA that I talked of, the formation of new virions and excretion. And we just felt that there was like no really realistic kind of like depiction of this. And Anne Liu from the University of Utah did an amazing job of doing that for us. Thomas Spletstößer also did this very nice animation of the virus. Now, you may be used more to this thing, which is the early depiction based on the SARS structures. We made this one. So what you can see is there's actually a stalk to the spike, which sticks out. The size of the spikes is huge compared to the rest of the virus. The heads of the lipids are actually smaller. And the number of proteins we show, as well as the size ratio, are based on averages that have been determined. I'm going to quickly stop the screen share in order to show you what we also did, which I think is very cool. So with help from Del Tronroth and Christopher Nolte and Matt Reeves, we did a model of the virus together with like this for comparison, which is a rhinovirus. So you can see it's really huge. And it's not round either, it's wobbly. And if you want one for yourself, you can, there are instructions on our homepage how to print your own virus model and how we painted it. And you can download the model files free from Thingiverse. So we felt that this might be great for education and outreach to actually like make the invisible enemy visible. So I just wanted to share this because, well, I think it's very cool.
Andrea, we are going to have to come to a close for that.
Yes, I only have 2 more slides. Right, this is the, oops, stop share. No, was correct. This is the inside of the virus, which Thomas Spletstößer also showed for us. And keep tuned, because soon we are going to have an entry animation of the virus on our homepage, how the spikes binds the virus and how the virus fuses with the host cell. We're going to release that in the coming like few weeks. We've also developed this together with the University of Utah. And in the end, I would like to say thanks to Thomas Splitstößer and Janet Iwas and Anne Liu, who have been illustrating for us, all the task force members and collaboration partners, everyone who's kind of like given input to this, because it's been like an incredible 8 months for us. We are funded by the DFG and the German Federal Ministry of Research and Education. We're now based at Hamburg University. And if you'd like to come to work for us and you're looking for a PhD student, or you know someone who's looking for a PhD in the area and would like to do methods developing, please make them write me an email to insightcorona@uniwurzburg.de. I'd be really glad to get their applications. And with that, I'm finished, and I'm very happy to get your questions.